Hello and welcome to the second longevity snapshot, where we will take a look at how exosomes can reverse cellular senescence and rejuvenate and increase the lifespans of mice. This video is presented to you by the Canadian Longevity Association, Canada's premier longevity nonprofit, and my name is Chris Linnell, the founder and president of the association. If you'd like to find out more about what we do, then please check out the links below this video. In addition, you'll also find a link to the study being reviewed here. And please don't forget to like this video and subscribe to our channel. So let's take a look at the study. In this study, researchers found a new way to deal with senescent cells. As a reminder, senescent cells are cells which have stopped dividing and have entered a dysfunctional state where they release all sorts of inflammatory molecules causing problems in the rest of the body. This has been a major area of aging research, and traditionally, two methods have been used to deal with them. One method, called senolytics, involves removing those cells. However, that can kill off other properly functioning cells. And the other method, called xenomorphics, involves keeping those cells alive but trying to reduce the inflammatory molecules they release. Here, the researchers have discovered a third method, which involves returning the senescent cells back to a fully functional state. They did this by restarting the cell cycle, whereby cells can once again divide and grow. To show you what I mean, let's take a look at the graphical abstract. So looking down here, you can see the cell cycle. Now, cells first grow in size, then they duplicate their DNA, grow some more, and then divide, and the process continues as the cells proliferate. In senescent cells, there's impaired DNA synthesis, which basically stops this growth cycle, causing the cells to become senescent. And the researchers found a way to restart that cell cycle and thereby rejuvenate the cells. They accomplished this by giving mice exosomes taken from human embryonic stem cells containing high levels of a microRNA called 302B. Exosomes are small spheres of chemical information that cells use to communicate with each other. And microRNAs are molecules which regulate gene expression. Without dwelling on this abstract, let's take a look at the paper itself. And they started off with cells in a dish. Looking at the study design here on the top left, they took fibroblast cells, which are connective tissue cells, and let them divide a number of times. Now, after 30 cell divisions, the cells have yet to become senescent, so those were the controls. However, after 50 cell divisions, the cells do become senescent, and they either gave them a salt solution or these exosomes taken from human embryonic stem cells. And you can see in this image here in B, on the left are the controls, in the middle are the salt solution cells after 50 divisions, and on the right are the exosome given cells. So this is a staining for senescence, and after 30 divisions, you can see there's no blue staining at all. After 50 divisions, the salt given cells have quite a bit of blue in them, indicating high levels of senescence, whereas the exosome given cells have much lower levels of blue, which can also be seen in this bar graph on the right, left being the controls, middle being the salt given cells, and on the right, the exosomes, and there are much lower levels of senescence in the exosome given cells. And in D here, looking at two markers of senescence, P16, P21, and 3 inflammatory markers, levels were much lower in the exosome given cells. So those are the green bars compared to the control red bars. And looking at G, how well the cells could proliferate, in other words, divide and grow. The top line shows the cells after 30 divisions, and you can see that they continue to grow and proliferate rapidly. The bottom line shows the older control cells, and as you can see, they don't really grow or proliferate that much, whereas the exosome cells in the middle proliferate considerably more than the controls, though not as well as the younger cells. And in H and I here, we see levels of Ki-67, which is a marker of proliferation, and the green bar is much higher than the red bar, once again showing that they could proliferate more, though once again not as high as the younger cells on the left. The bottom here shows differences in gene expression between the exosome cells and the control cells. Next, briefly looking at single cell resolution. And I'll skip over most of this, but I just wanted to show you differences in the cell cycle between the salt-given cells and the exosome-given cells. And as you can see, this light blue section of these bar graphs is higher in the exosome-given cells. And that is part of the cell cycle where the cell copies its DNA. So this is also a marker of proliferative ability. The analysis here shows that the cells could proliferate once again. Now we move on to mice. They used 20-month-old mice, which is roughly the equivalent of a 60 to 70-year-old human, and they gave them weekly injections of either salt solution or the exosomes, tested them at 30 months, and then let them live out their lifespans. And as you can see in B on the right, the exosome-given mice lived longer. Now, in all of these graphs, green means the exosome-given mice, and red means the salt solution-given mice. And the exosome-given mice lived on average 5 months longer, which is an 18% increase. Looking at their weights in C here over the first 10 months of the study, you can see that whereas the control mice continued to lose weight, the exosome-given mice actually increased their weight. And considering the fact that mice tend to lose weight as they grow older, this is a positive development. Next, they looked at their physical performance. And in D here, the experiment was how long mice could stay on a rotating rod, which increased its rotational speed over the course of 300 seconds. And the exosome-given mice managed to stay on this rod considerably longer. In a second similar experiment, where the rotating rod had a fixed speed of 25 rotations per minute, once again, the exosome-given mice managed to stay on this rod considerably longer than the control mice. Their grip strength was also significantly higher compared to the control mice. Next, to test their cognitive abilities, the mice were placed in a bowl of water, I guess, and they had to try to find a sunken platform to sort of get out of the water. And the mice were placed in quadrant 1, with the sunken platform being in quadrant 3. And the control mice swam quite a bit around to try to find this platform, whereas the exosome-given mice found the platform more quickly, as can also be seen by this graph in H here. So G and H was the training phase, and the exosome-given mice learned to find that platform much more quickly. And in I and J was a memory test. So the mice were once again placed in the water. However, there wasn't a sunken platform. So the mice were tested to see how well they remembered where the sunken platform should have been. And in these heat maps, whereas the control mice swam all over the bowl of water, the exosome-given mice swam mainly in the area where the sunken platform had been, which can also be seen in the bar graph in J here, where again the sunken platform was in quadrant 3, and the exosome mice swam the majority of their time there. Next, looking at inflammatory markers in K. And we have young mice on the left, the control old mice in the middle, and the exosome old mice on the right. And for all 4 of these inflammatory molecules, levels were much lower in the exosome-given mice. In L, we're looking at senescence once again. And as you can see, the young mice have little or no blue staining, showing low levels of senescence. The aged control mice have high levels of blue, indicating lots of senescence, whereas the aged exosome mice have much lower levels of this blue staining. And the same can be seen in these bar graphs to the right in the various organs: kidney, liver, lung, spleen, and skin. And once again, the red bar is much higher than the green bar. The green bar is not as low as the young controls. Next, the researchers looked at individual cells taken from these mice. And we'll just briefly look at this, starting with A in the top left. So cells taken from either the liver or the skin between the old control, the old exosome, and the young mice. And this is the cell cycle. So once again, S is when the cells copy their DNA, and the length of time spent in the S phase was longer in the exosome-given mice, comparable to that of the younger mice, both in the liver and in the skin. And looking at F in the bottom right here, once again in the liver and skin, and these are markers of either senescence, so P21 or CCNG2, and markers of proliferation, that KI67 again. And here the light blue are the old controls, the red are the exosome mice, black are the younger mice. And in each of these cell types, the exosome-given mice were much closer to the young mice than the old controls, as can be seen by the fact the red bars are much closer to the black bars as opposed to the light blue bars for each of these. Next, the researchers wanted to see what was causing this. And again, without going into too much detail, taking a look at A in the top left. So this shows levels of various microRNAs in the cells, and they ranked them according to their abundance, or how much of these microRNAs were in the cells. And the microRNA which had the highest levels was called 302B, and it was considerably higher than any other of the microRNAs. And this microRNA has a role in cellular reprogramming and maintaining the stemness of, stem cells. And when given on its own, this microRNA 302B, and I'll just call it 302B from now on, significantly reduced the levels of senescence in cells that were given it, as can be seen by this lower bar graph on the right compared to this one here on the left. Next, they wanted to see what would happen if they gave exosomes containing high levels of 302B to mice. And this time they didn't take exosomes from human embryonic stem cells. They just took exosomes from regular cells and basically managed to increase the levels of 302B inside them before giving them to the mice. And in this experiment, they started with 25-month-old mice and followed them until they were 30-month-old, at which point they were terminated and analyzed. And I should mention that they use black 6 mice, which are just normal mice, the most common mouse used in experiment. Now let's make this a bit bigger so that you can see. And in C here, you can see the visual differences between the mice. So on the left are the aging control mice from 25 to 30 months, and on the right are the 302B given mice. And you can see that the mice are starting to lose their fur on their backs, which got worse in the control mice. However, for the mice given exosomes from just before they were given the exosomes to 5 months later, you can see that actually their fur regrew. And next, looking at weight, and rather annoyingly, the authors have flipped the colors around. So this time the control aging mice are in green and the exosome given mice are in orange. And you can see that the 302B given mice maintained their weight much better than the controls. The time they managed to hang on to a rotating rod also increased, reaching high levels of statistical significance, as can be seen by the stars above here. The more stars indicating greater statistical significance. And just like this rotor rod test, their grip strength also got better compared to the control mice, whose grip strength declined throughout this period. Moving on to cognitive abilities. Once again, the mice were trained to find a sunken platform in a pool of water. And at the top here are the controls, the bottom are the 302B mice. And at the start of the study, both sets of mice swam all over the place to try to find the platform in the third quadrant. However, 5 months later, whereas the control mice continued to swim around everywhere, the 302B mice swam almost directly to that platform. And you can see in this bar graph, the time taken to find that platform was considerably less as time went on compared to the controls here. On to the memory portion. At the beginning, both controls and treated mice swam around everywhere looking for the platform that was no longer there. However, 5 months later, whereas the control mice still swam around everywhere looking for the platform, the treated mice stayed mainly in the third quadrant. And again, these are sort of heat maps showing the amount of time they spent in a certain area. And this can also be seen in the bar graph, which shows how much time they spent in that third quadrant. And once again, it was considerably higher than the control mice as time went on. Didn't really change much with the control mice. They spent longer amounts of time in that quadrant over the course of the 5 months. Next, they looked at inflammation and senescence. So starting with inflammation and looking at these 4 inflammatory markers in eye, for each of them, levels were much lower in the treated mice than in the controls, and it reached high levels of statistical significance. Looking at senescence, on the left here are stained cross-sections of these various organs, and the blue staining indicates senescence, and in the control mice, there's a lot of blue. However, in the treated mice, there is much less blue, as can also be seen in this bar graph, again with the 5 different organs, and the orange bars are significantly lower than the green bars, again showing that there is a lot less senescence in these organs. Finally, they looked at other markers of senescence and proliferation, once again in brain, liver, and skin. And just a reminder, P21 and CCNG2 are markers of senescence, and Ki67 is a marker of proliferation. And in each of these, the markers of senescence were lower in the treated mice, and the markers of proliferation were higher in all 3 organs. So altogether, these experiments showed that this treatment improved their physical appearance, their performance, their cognitive abilities, as well as lowering levels of inflammation and senescence. However, what about their lifespan, since these mice were euthanized at 30 months of age? Well, that is exactly what these researchers looked at next. So starting with the study design, and I'll make it bigger once again, instead of using 25-month-old mice, they went back to using 20-month-old mice. So once again, that's between the age of 60 and 70 in human years. And every 2 weeks, they gave the mice either a salt solution or these enhanced exosomes containing higher levels of 302B. And they continued giving them the treatment until all the mice had died. One thing I really liked about this experiment was that a relatively large number of mice were used, 80 in each group. Now, quite often in these types of experiments, only a small number of mice are used, which means that it's harder to reach statistical significance. In addition, an equal number of male and female mice were used. Once again, quite often only one sex is used due to cost reasons, and therefore it is impossible to know if there are any differences in lifespan according to sex. Before looking at lifespan, let's see how their body weight changed over the course of the study. So in these graphs, the red represents the exosome 302B mice, the blue represents the controls, and the treated mice maintain their body weight much better than the controls. Moving on to lifespan now, and starting with the combined male and female mice, you can see that the treated mice lived considerably longer than the controls, as we can see by the survival curves. In fact, the median lifespan, or the average lifespan, was 15% greater. Looking at the sex differences, starting with the male mice, the treated mice lived on average 22% longer than the controls. As for the female mice, they lived on average 14.9% more than the controls. Looking at maximum lifespan, in D here, which was when 15% of the mice were still alive. In the combined group, maximum lifespan increased by 12.13%. For the men, it increased by 9.86%, and for the females, it increased by 13.98%. So both for average and maximum lifespan, these are quite impressive results. However, one potential issue with reversing senescence and allowing the cells to proliferate again or divide again is the fear that this might cause cancer, because after all, cancer cells proliferate. And this is true especially in mice, since they tend to die of cancer if they live out their natural lives. What the researchers found, however, was that there was no real difference between the control and the treated mice. There was no significant difference at all, as you can see by these 2 bar graphs here. Likewise, when they came to look at the number of diseases a mouse had when it died, there was no difference between the controls and the treated mice. So although they lived longer, they seemed to die of the same causes. And when looking at the different types of tumors the mice had, there was once again no real difference. I mean, maybe the treated mice didn't have any sarcoma, maybe they had a bit more lymphoma, but overall there was no statistical significant difference between the control and the treated mice. And at the bottom here, this simply shows that in the 4 organs, liver, kidney, lung, and skin, they showed that the treated mice had restored hallmarks of proliferation and reduced hallmarks of inflammation. So what does this all mean? Well, firstly, the researchers have shown that there is now a third means of combating the negative effects of senescent cells, not by getting rid of them, not by reducing their inflammatory markers, but by restoring the function of the cells and restoring them to a more youthful state. It also once again shows the power of exosomes, which I believe is an underappreciated avenue for tackling aging and provides us with a promising avenue for longer and healthier lives. I hope you will agree with me that this was an exciting study. However, I would love to hear your thoughts, so please share them in the comment section below. And that marks the end of another longevity snapshot. Please don't forget to like this video and subscribe to our channel. So until next week, goodbye.